Hi everyone, welcome to part 2 of this video series on a summary of the integumentary system, whereby we highlight the most important concepts. In part 1 we labeled the diagram. Here's an example of the diagram that we labeled. So please refer to part 1 whereby we label this diagram. You should know the various structures, and if a similar diagram appears on the test, you should be able to label it. Let's go through a couple more slides to highlight a few other important concepts. This particular slide shows you the layers of the epidermis. So the layers of the epidermis are the stratum going from superficial to deep, stratum corneum. This is the dark layer over here. You can see this is the dark layer. A darker layer, that is the stratum granulo. So this is the stratum spinosum, and these cuboidal cells right at the bottom of the epidermis is the stratum mucillae, or the basal layers. So those are the 4 layers. Not shown is stratum lucidum, which is only found in thick skin. And when we say thick and thin skin, is just relatively speaking. Thin skin is half a millimeter thick and is found all throughout your body. That's thin skin. Thick skin, on the other hand, is really about 3 times as thick. It's 1.5 millimeters thick, and it is found on the soles of your feet and your palms of your hand, for example. So that's thick skin. This obviously is thin skin, since there are only 4 layers for the epidermis. The epidermis is stained more deeply than the dermis. Here's the dermis. All this is the dermis. And it's still deeper than that. This particular structure is the dermal papillae, whereby the dermis invaginates into the epidermis. Let's talk about the layers of the epidermis in more detail. All the cells in the epidermis are what we term keratinocytes, because they secrete keratin on the surface of the stratum corneum. You will see keratin, which are these little swirls. We'll see that in another photomicrograph. So these are all keratinocytes. In the basal layer, these cells are active. They're cuboidal looking, as you can see. These cuboidal cells in the basal layer, they are active, mitotically active, and they constantly are reproducing. As the new keratinocytes are produced, they slowly migrate upwards, or more superficially, towards the stratum corneum. In the process, these keratinocytes lose their nucleus, and by the time they reach the stratum corneum, they're actually dead cells over there. So these are dead keratinocytes in the stratum corneum, and those cells will be shed. That's our skin shedding, only to be replaced by the new layers of these keratinocytes that are migrating towards the stratum corneum. Right, so that's how epithelialization occurs. So whenever we have a defect or incision or any scrape on our skin, it will re-epithelialize, and that's quite a handful. So the epithelium will regenerate itself, because it continuously is dividing, and that's because of these mitotically active cells in the basal layer. So these are all keratinocytes over here. Let's move ahead. Oh, by the way, what's the tissue? The dermis has 2 layers. Firstly, the most superficial layer is the papillary layer, and the deeper layer is the reticular layer. What's the tissue in the dermis? It's mainly dense, irregular connective tissue, but as you can see in the papillary layer, which is what we're looking at over here, it is more loose, so to speak. So if you put down loose areolar connective tissue, you probably wouldn't be incorrect either. So, but as we go deeper into the dermis, it does become more denser, and hence it's really more appropriately labeled as dense irregular connective tissue. So here we can see the relative thickness of the dermis to the epidermis, right? So in the previous diagram it wasn't readily apparent, but here we can see the integumentary system is mainly the dermis. The epidermis makes up a tiny fraction of the superficial layer. So all this dark part over here, that's the epidermis, and everything else is the dermis. So in terms of layers of the epidermis, of the dermis, this is the papillary layer, and the rest of it is the reticular layer. Again, there's no hard boundary between the papillary and reticular layers. Alright, one last thing I want to point out is here we can see the actual keratin on the surface of the stratum corneum. The keratin secreted by the keratinocytes, which is the main cell in the epidermis, makes the skin tough and provides its waterproof layer. So here we again have the different layers of the skin of the epidermis. More specifically, SC stands for stratum corneum, SGR stands for stratum granulosum, SS stands for stratum spinosum, and SB stands for stratum basilae, or the basal layer. This is the dermal papillae, and you can see here PL stands for the papillary layer, and RL stands for the reticular layer. Obviously EPI is the epidermis. I guess if I just write dermis, that will imply that. So EPI is really the epidermis, and D is the dermis. Oh, no, not there. That's the derm part over here. That's dermis over there. The D over here stands for duct. This is a duct that is coming out from the dermis all the way into the epidermis. Now, what duct that is, that could be typically it would be a eccrine sweat gland, simply because they're more eccrine sweat glands than they are, say, apocrine sweat glands, but it would depend. But in either case, you could see it's the duct. Also, the staining, you can see it's darkly stained over here. That tells you that all the appendages are in the dermis, such as the eccrine sweat gland, the receptors, and so forth. Those are all appendages of the epidermis, really, because these are epithelial cells as opposed to connective tissue. So that's why even the duct over here, which will be simple cuboidal cells, or stratified cuboidal if this were a sweat gland, an eccrine sweat gland, these are all epithelial cells that are obviously stained darkly. And all the cells in the epidermis are epithelial cells, that's why they're stained darkly, as opposed to the tissue in the dermis, which is loose, which is connective tissue, and that's not stained very well. Right? The pink stuff is the collagen fibers, and you see some nuclei, and so forth. We did the differences between epithelial tissue and connective tissue in chapter 4. Let's move on and see here. So what layer is this? What SS stands for? Obviously stratum spinosum. And it's so called stratum spinosum because you can see the spiny projections. Can we actually see them here? Yeah, sort of. Like you can see these little spiny projections. I guess you have to use your little imaginations, right? So that's how it It's got its term because they are spiny projections as the keratinocytes migrate from the SB, the stratum basilae, towards the stratum corneum. Here in the layer just superficial to the stratum basilae, it gets the stratum spinosum. The keratinocytes here have little spiny projections. That's how it got its name over there. Again, CT, connective tissue, that's the dermis. Here are two photomicrographs, and can you tell what the difference between these two photomicrographs of the integumentary system are, with specific focus obviously on the epithelium of the epidermis itself, right? So I'll ask you to focus on the dark layers over here, this pigmented, maybe you can switch to a highlighter. Yeah, all this part over here. What's all this really dark colored stuff? As opposed to here, where you don't really see that, right? What's that? If you replied that's melanin in the melanosomes that are secreted by the melanocytes, you'd be correct. So this is dark skin, and this is light skin. The melanocytes are specialized cells in the basal layer that secrete melanin. And melanin is a photopigment that protects us from the harmful UV radiation, right? So if you live in tropical climates closer to the it's NIN. If you live in tropical climates or closer to the equator where the sun is high in the sky and you need more melanin in order to protect yourself from the ultraviolet radiation, hence the skin tone is darker. If you live in more northern latitudes where the sun is typically lower down in the sky, especially in the winter, well, in that case you don't need to secrete as much melanin, and because there's not much sunlight, and you'll end up with a lighter skin tone. Interestingly, though, in terms of irrespective of whether you have light skin or dark skin, we all have the same number of melanocytes. What's the difference is the amount of melanin that we have, that is really darkly stained over here in dark skin that you can see. So we produce the same amount, we have the same number of melanocytes, but we produce differing amounts of melanin. Right? That's the difference between light skin and dark skin. There are other photopigments. So melanin is really a photopigment. There are other photopigments in the skin. So some folks have a more reddish tone, so they have more of the keratins as a photopigments and so forth. So we're not going to talk about that, but we'll just stick with melanin as the main photopigment, which it is over there. So melanin is secreted by the melanocytes, and then it goes into melanosomes. And really, for someone with a darker skin tone, they have bigger melanosomes because there's more melanin in the melanosomes. So it's like an endosome, right? So remember, in the cell, everything needs to be packaged up, so we call it a some. So endosome is just something that has some stuff inside it. So melanin is a polymer, so you can't just have it floating around in the cells, so it's packaged into melanosomes. If you have dark skins, the melanosomes are bigger because they have more melanin, and if you have light skin, your melanosomes are smaller because they have less melanin. So that's the difference between light and dark skin. Let's move ahead. Let's just move ahead over here. What's the difference between these two skin? It's roughly at the same magnification. So I'd ask you to look at the epidermis. So the epidermis over here and the epidermis over here, it's actually at the same level of magnification. What's different is this layer over here, which is the stratum corneum. The stratum corneum is much thicker over here, and the stratum corneum is much thinner on the right side. So this is right, that's left. over there. So this is thick skin. This is thin skin. We find thin skin all throughout the body. Thick skin is only found in a couple of locations, the palms of our hand, the soles of our feet. And why we say thick or thin, it really has to do with the thickness of the stratum corneum over here. You can see the stratum corneum is really thickened. It has multiple layers over here in thick skin, and it's not as thick, obviously, in thin skin. The rest of it is the dermis over there, and we'll go through the structures later on. The HF stands for a hair follicle. So here, this again, let's go through the layers of the epidermis. That's the stratum corneum, very thick. So yes, this is thick skin. These are dead keratinocytes. Then we have the stratum lucidum. We haven't seen that layer before. And the stratum lucidum is the fifth layer of the epidermis that is only found in thick skin. And this is certainly thick skin from your soles of your feet or the palm of your hand. And then we have this layer over here, the darkly pigmented layer. That's the stratum granulosum. Oops. Stratum spinosum. And then we have the basal layer, stratum basilae. Right, so make sure you know the layers of the epidermis. In thick skin, 5 layers. In thin skin, 4 layers. You should be able to recite it going superficial to deep, stratum corneum, granulosum, spinosum, and basilae in thin skin. And yes, we have stratum lucidum in thick skin. Or going from deep to superficial, stratum basilae, spinosum, granulosum, corneum. Right, so make sure you do that. Don't forget the stratum. Okay, what's this MC? The MC stands for the Meissner corpuscle. In the previous video, we talked about various mechanoreceptor or touch receptors. So I'm not going to go through it over here. I'll ask you to refer back to the earlier video, part 1. But essentially there are 4 types of touch receptor: the Merkel cells or discs that are actually found in the epidermis. And that's for, these are slow adapting mechanoreceptors for light touch, so it detects a sustained touch on the skin. Then we have these Meissner corpuscles. So let's just put down over here. This is rapidly adapting fine touch. So they detect moving touch, so to speak. So if someone's just literally rubbing against your skin, you still feel it as it's being rubbed against. As say if someone else's finger is moving across your skin, that will be the Meissner corpuscles detecting it, because they are rapidly adapting over there. So sorry, that will be the Merkel cells that will be detecting that, right? So the Merkel cells are slow adapting. So it's sustained touch on the skins. The Meissner corpuscles are rapidly adapting. So if someone just literally just touches you, and then the Meissner corpuscles will detect it and immediately forget about it. So again, more detail is there. So this is slow adapting. More details in the previous video. This is fast adapting. Both of them for fine touch. And then we have the deep touch receptors, 3 and 4, which are the Ruffini, Ruffini endings. Slow adapting for more steady pressure, so they'll detect steady pressure on the skin. So we're not talking about just a gentle touch, but a more sustained push down on the skin. And since they're slow adapting, they will detect it. Hence, it's really about skin stretching. So anytime your skin is stretching, it's the Ruffini endings that are detecting it. So that's deep touch, both these receptors. And lastly, we have the Pacinian corpuscles, which are rapidly adapting deep touch. So if someone's just pushes down deeply against your skin, the Pacinian corpuscle will detect that rapidly, and they're rapidly adapting, so it will dissipate quickly as well. Alright, so we have these touch receptors over there. The MC is the mycin corpuscles, which you can see are located in the dermal papillae. So they make sense because they are for fine touch. They're close to the epidermis, basically touching the epidermis. So make sure you go over the receptors, the 4 touch receptors. We also have thermoreceptors for hot and cold, and we have pain receptors, which are the free nerve endings, also referred to as nociceptors. We'll talk more about the nociceptors in year 2, hence focus on the touch receptors and the thermoreceptors. By the way, the Ruffini endings are also thermoreceptors. They detect heat. So that's it for chapter 5. Next class we'll do chapter 6. That will be after the test. Actually, one last thing I just want to point out in terms of cells. So we talked about the cells, which are the touch receptors, and the nerve endings are the thermoreceptors, except for the Ruffini endings, which are also thermoreceptors, but they're not nerve endings. It is a nerve ending, except it's encapsulated. We're talking about free nerve endings. So these are pain receptors. Both of these are normally free nerve endings. And pain receptors are also called nociceptors. There are other cells that I also want to talk about in the integumentary system, and they are the Langerhans cells, or the macrophages. Langerhans cells are the resident macrophages in the integumentary system. So if you have a break in the skin, various pathogenic bacteria and viruses can obviously make its way into the body now, and before it reaches the bloodstream where it can disseminate throughout the body, and that will be a really bad thing, you want to detect those foreign pathogens and then activate the immune system. And that's what the Langerhans cells do. They are immune cells involved in immunosurveillance. We'll talk more about the immune cells in ANP2, but the Langerhans cells, as I mentioned, are a form of the resident macrophages in the dermis of the skin, and the macrophages are also phagocytes. So we'll talk about phagocytes in more detail in the immune system. The last thing that we're going to do is to describe very, very briefly the physiological functions of the skin. This is covered in the textbook, and essentially you should revise that on your own. We have protection as the first function. So the skin acts as a physical barrier to foreign pathogens and the external environment. It also acts as a chemical barrier because skin secretions are manifold. So lysozymes are secreted. The skin also secretes acid. There's an acid mantle that helps to inhibit the reproduction of bacteria, for example. In addition, you can think of melanin that provides protection as well. Melanin is there to protect against the UVA and UVB rays. So that's all about protection. We also have biological barriers, so the natural. So maybe we should write this down. So this is the physical barriers, chemical barriers, lysozymes and the acidic mantle. Then we also have biological barriers. That's the natural flora on the skin, we have a whole host of symbiotic bacteria that live on our skin, and those symbiotic bacteria are not harmful, and in fact they serve a biological function in that they crowd out all the bad bacteria. So we have so much good bacteria on our skin essentially that there's no space for the bad bacteria to grow. The good bacteria outcompete all the bad bacteria. So that's the biological barrier. The second physiological function is thermoregulation. We have obviously the eccrine sweat glands that play a very important role. A third physiological function of the skin would be cutaneous sensation. We talked about the various touch receptors, which are the mechanoreceptors. We even have thermoreceptors, and we have pain receptors. For example, the fourth physiological function are the metabolic functions that the skin provides. So essentially that would be vitamin D synthesis, and this is where the melanin also kind of plays a role. Melanin acts as a chemical barrier over here, but we know melanin helps to protect against UVA and UVB rays, but we also need UVA and UVB rays in order to synthesize vitamin D. So it's a delicate balance over there. Too much melanin, then you're not going to be able to synthesize vitamin D. Too little melanin, and then UVA and B radiation can penetrate the skin and cause various mutations, and perhaps even lead to skin cancer, for example. So that's the metabolic function. And as long as you know about the role that vitamin D plays, that should be enough. Vitamin D is essentially necessary in order to absorb calcium as well. The fifth physiological function, to go back over here, is to act as a blood reservoir. So when core body temperature drops, for example, all the blood, or a lot of blood from the skin, is diverted to the critical internal organs in the body. So blood is diverted from the skin to the viscera, where you keep the vital organs running, right? But you can also think of this particular physiological function of the skin and working hand in hand with thermoregulation. So when core body temperature rises, then more blood will be diverted to the skin in order to dissipate the heat from the blood. And when core body temperature drops, as I mentioned, blood will be diverted away from the skin in order to sustain the vital organs. The sixth physiological function of the skin is to act as it plays a role in excretion. So for example, sweat contains some urea and various other ammonia byproducts, and in that way we're getting rid of excretion over there. So this protection, I think the book lists 7, but here's 6. You can subdivide the first physiological function in terms of protection. You can have biological barriers or chemical barriers, whichever way you want to classify it doesn't matter, but these are the basic physiological functions of the skin. Please make sure that you do revise them in order to answer any question that might be relevant for the test itself. All right, so that's really the only physiological part in terms of chapter 5. All the rest is more or less anatomy and some short answer questions as well that will be related to the various, for example, cells in the skin, the various layers of the epidermis, etc. So label the diagrams in terms of the anatomy, and then go over all these other short answer type questions in terms of the cells and whatever else we covered over here. And lastly, finish off by revising through the physiological functions of the integumentary system. That's it for chapter 5. Thanks. Have a great day. Bye.